The overall goal of this procedure is to detect and quantify atherosclerotic plaque activity and vascular inflammation in medium and large human arteries. Start with a whole-body FDG-PET CT scan and image the aorta and its major branches from the heart down to the feet. Then qualitatively analyze the images for presence of vascular inflammation. Using the registered CT scan, quantify the amount of radiotracer detected at each slice of the PET scan and localize regions of interest. Proceed to calculate the standard and novel outcomes that measure vascular inflammation. Ultimately, the results depict and quantify the burden of atherosclerosis in any given arterial region through the utilization of FDG-PET CT with quantification.
FDG-PET CT can help answer key questions in the vascular imaging field, such as how does therapy modulate plaque activity, or how does vascular inflammation change over time in vivo. The implications also extend toward diagnosis of subclinical atherosclerosis in vivo, because current techniques do not allow for whole-body real-time molecular imaging that can be used for response assessment purposes in a short-term period.
FDG-PET CT can provide insight into the natural history of atherosclerosis because it can also be applied in the setting of diagnosis, therapeutics, and prognosis.
There are several advantages of FDG-PET CT over existing methods like CT angiography or coronary artery calcium scoring by CT. With minimal operator dependence, FDG-PET CT affords high contrast resolution with real-time in vivo whole-body molecular imaging. Quantification of plaque volume with metabolic activity allow for multimodal atherosclerotic plaque quantification. These factors together facilitate ability